All right, we are recording. Before we start, we do want to provide a land acknowledgement. We respectfully acknowledge the territory in which we gather as the ancestral homeland of the B.SC and the island of Newfoundland as the ancestral homeland of the MIGS and B.SC. We would also like to recognize the Inuit of Nunavut and Nunavut and the Innu of Netsima and their ancestors as the original people of Labrador. We strive for respectful relationships with all the people of this province as we search for collective healing and true reconciliation and honor this beautiful land together. Now, join me in welcoming today's seminar speaker, Ben Grady, is here with us from AMAR Seafood, is that? Yeah. In Victoria, Prince Edward Island, where he's the hatchery manager at the facility. Ben graduated from Memorial University with a B.SC in biology, focusing on the ecology and conservation. He now specializes in the rearing of halibut from egg to the larval stage at North America's only facility to house the complete life cycle of the Atlantic Halibut. With that, I'll hand it over to you, Ben. If you can.
Yeah. Hopefully. See how easy it is to share this.
Yeah.
Is that working all right?
Yeah, looks good on our end in the hall.
Wonderful. Okay, so this is a unique look at aquaculture. Oh, I've been changing Dropbox. Okay. We do Atlantic Halibut and Spotted Wolffish in Victoria, Prince Edward Island. Just to give you all a little overview of what we're going to talk about today. I put a little bit about me, but I think that was already covered, so we'll probably just like skip through that quick. Little introduction to AMAR Seafood, a bit of history about how we got to where we are right now. A little bit about our facility and what we have here. The production of halibut at various stages. So we'll start at the brood stock and kind of work our way through the life cycle. And the future of wolffish aquaculture here. It's gotten started, but there's definitely still more to come. And then I have a couple exciting opportunities that I've gotten to do through work that I'll just mention at the end to kind of show that aquaculture as a career isn't always just like feeding fish and cleaning up after them. Yeah, I guess the only thing new here is that I started working in aquaculture in 2018 with Halibut PEI, which is the company that was previously located in this building. They went bankrupt January of this past year. And in 2019, I started running the hatchery here. So we're AMAR Seafood. We're located in Victoria, Prince Edward Island. The company was officially established in June of 2022 in the old Halibut PEI building. We kept all the brood stock and the nursery fish that we had in between the closing of Halibut PEI and the opening of AMAR. I worked here by myself for 6 months, so I kind of branched out from being just the hatchery manager to having a little bit of experience with everything that goes on. But right now the facility supports brood stock through to harvest for Atlantic halibut and also spotted wolffish. There's 5 people currently employed on site, but we're definitely looking to expand in the very near future. So this is Scott Travers on the left. He's our CEO. and he was the CEO before we went out of business with Halibut PEI as well. He's been around since I've started, along with Dr. Gerald Johnson on the right. He is our director of science, again, has been for as long as I've been around. He's a vet pathologist by trade, so he brings a lot of knowledge to the table in the director of science role. And then around May of this year, we had our first visit from the AMAR group. So that's Bjorn Apeland on the left and Knut Trelvik on the right. They're the CEO of the AMAR group and the president of AMAR North America. I think I got that title correct. So they came in May, toured the facility for the first time, and as of June of this year, the building became the home of AMAR Seafood, and they've brought a lot of experience and knowledge and opportunities for us in the branching out into wolffish. That was kind of their thing that they had started already, and they were looking for a bigger facility for it to kind of take hold, and we have what they need. So that's where we're at now. I'm just gonna move this little window. Sorry about that. So that's a picture of our facility. It's nothing special on the outside. There's a lot going on indoors though. It used to be a lobster pound once upon a time, and it's been expanded and converted since then into an aquaculture facility. So we run solely on land-based aquaculture. We don't do sea cages. The bay is too shallow when the tide goes out, and there's too much uncontrollability on the straight side to really consider it, but it's kind of been part of our mission since we started to focus on land-based and not really get into sea cages. They provide greater control parameters such as oxygen and temperature. We're not just like having random water flow in, it's all tank-based and comes from our wells. So we have more control over what the temperature is going to be through chillers and different, like we can add oxygen or take oxygen, or well, not take oxygen out, but if we need to add oxygen, we will. It has a limited environmental impact. So all of our effluent that we have is treated before it goes out into the strait, I guess, is where it exits. Then we don't have any kind of, we have like a settling tank that kind of stops the more like the waste solids, waste feed that exits. So we have monthly water samples that get sent into the government to show that our total suspended solids, our nitrogen and our ammonias are all controlled properly. The only time we ever had an issue with it was one time somebody stuck their finger in the bottle, and our total suspended solid count got so high that they like called us right away and they were like, you might have to shut down. But we just sent another one in and it was okay. We also have no impact from predation. So with sea cages, there's a lot of opportunity for wildlife to kind of get at your stock. Where we're all indoors, we get, you know, a random bird every now and then will make its way into the facility, but there isn't really any risk of predation inside. It does require filtration to control the contaminants, both within the facility and as the water exits. We require a source of water to do that, to run the facility, which we have in our wells, which I'll talk about after this. And it is a little bit more expensive to run because, you know, you have to pump your water. It's not just free flowing through the sea cages like you would have there. And with our recirculation system that we have, again, which I'll talk about later, there's a lot of expense in running those massive pumps that are required to run it. Maybe. There we go. So we have 3 saltwater wells on site. They're 200, 150, and 400 feet deep. Each of the wells is accesses a separate aquifer, so it has its own temperature profile and its own salinity profile as well. Because of the natural filtration of the groundwater into the wells, we're free of pathogens. So that allows us to grow our fish free of antibiotic use, which is really great. We got ocean-wise status for our treatment of the fish for when we harvest and sell our product. So these are some monthly temperature graphs over the years. You can see that the nursery tank, that's well 1, it fluctuates quite a bit. The grow out kind of stays a little bit more, that's our 3rd well. And then our heat recovery system is a way that we control the temperature in the system. And that's usually when the water gets too cold for the fish. They prefer it to be like a 10 to 12 degree water. And we can add warmer water to the tanks if we need to keep their feeding up and their physiology functioning properly. Here's one from 2020. So you can see kind of the same thing year to year. We're getting the same kind of profile. And 2021, with the climate getting a little different, we're having a little bit more of like a unpredictable curve of our temperatures, but the trend tends to stay the same, of the low in May and high in October, November. Two, three, four, five of our different sections of the building run on recirculating aquaculture systems, or RAS. So this is an example of one of them. This is our first feed room. And I just have some pictures to kind of go through how the system works. I'll leave it up for a second just so people can see and take in the majesty of everything that it is. So we've got our head tank. This is where the water starts before it flows into the tanks. You can see the inlet on the right side of the tank, and then the drain is actually at the bottom in that little odd shaped rectangle. The water flows from there into our sump tanks, which is just where like the dirty water ends up, and where we take it for filtration. We've got our propeller bead filter, which uses a bunch of small like plastic beads to kind of pick up solids that are in the water and then flush it out. The beads inside have to be cleaned every season, just to like make sure, at least every season, just to make sure that we're not like carrying over contaminants from the season before that might end up getting washed through. We've got our foam fractionator. So this is a protein skimming unit. It also tends to take out a lot of our clay that we use in the tanks for shading. It's pretty disgusting, but it's cleaned daily to make sure that it remains tidy. It works by forcing water up the center pipe with some air, creates a foam, and proteins kind of adhere to the bubbles, and then they spill out over the top. And then water is like returned back into the tank. Over here we have like our primary filtration loop. The other 2 are more auxiliary filtration units. We've got our pumps at the bottom. They flow through a helix filter, which you can see at the bottom here. At the top is our UV, so the UV kills bacteria and viruses, that sort of thing. The helix filter there, that's the filter. It's a little bit dirty in the picture, but it takes out most of our live feed that's uneaten, or when we're weaning, it grabs the pellets that are left over. This is our Bubba filter. It takes out most of the clay that we use for the shading. So when the larvae are in the tanks, we have 24-hour light on them so that they're eating constantly. The shading from the clay provides a level of turbidity to the water that keeps their stress levels low. And it also allows them to, the turbidity allows allows them to see the live feed that's in the tank easier, and lets them to hunt better, essentially, so they can see the movement closer to them rather than having to focus the whole distance across the tank. That's what the Bubba filter looks like. It's essentially the same as a hot tub filter. I think we actually buy them from a company that sells hot tub filters. And then, yeah, this is just the overall view of the tank. I didn't actually show you guys the parabolic filter on the right. It's that that white box with the handle on the top. That collects bigger solids before the water flows into the sump, just so that we're not getting large solids clogging up our filtration system. Supervisory control and data acquisition. This is our SCADA system. We use it to monitor and control various set points and parameters across the facility. We can examine trends in temperatures and oxygens, salinities, anything else that we kind of measure throughout the facility. We can use it to set and control instrumentation. So we have auto feeders that used to be set up. We just got new ones, so we're working on bringing their functionality into the system right now. But we used to be able to, with our old feeders, adjust the amount of feed that they would be getting from SCADA, so you could do that remotely from anywhere you had an internet connection. It also runs our alarm system and our callouts. So anytime one of the parameters goes outside of our control points, it'll actually send out a call to myself, and I can respond if possible on the internet, so that I don't have to go in to the facility. But if I have to go in, I know that I can go, and it'll tell me that there's something wrong. And we also have full customization of our pages and of the reports that we take out of it. So I'll show you a few of our pages that we have that are all kind of set up to fit our needs. This program can actually be used for a variety of different things. We just find that it works really well for us with our controls and such. So this is our nursery heat recovery system. It's pretty hard to explain the diagram. Excuse me, holy smokes, I'm burping like crazy. It's kind of hard to explain the diagram that we have here. It's a lot easier when we're downstairs looking at it, but I didn't have any pictures to put up. But essentially, you can see the yellow 28 that's highlighted at the top here. That's our control point, so we can adjust that to chill the water down to whatever temperature we want it to be at. 28 is in Fahrenheit, as well as the 40 and the 30 degrees there. We can also turn on and off the nursery heat recovery system remotely with the start and stop buttons. which is really convenient because when the power goes out, it doesn't turn back on by itself. So we can flip that back on before we're coming, like as we come back to the building, rather than waiting until we get back, because the temperature will rise up to about 14 degrees in most of the tanks without this running. This is the main hatchery dashboard. This is where I spend a majority of my job, is just looking at this page. You can see that we monitor our water temperatures, the air temperatures in the room. All of our pump frequencies and head tank levels are monitored. A lot of our systems are down right now because we don't have any eggs coming through the system just yet. The season doesn't start until November, mid-November. So there's a lot of red on here that shows that things aren't working, but that's just because they're off. It's not like, this wasn't a terrifying screenshot to take. And then we have spots set up for measuring salinity and total gas pressure, CO2, pH, and oxygen. We're only really using the total gas pressure right now because the other probes that we had weren't great. They weren't like measuring consistently or very nice. So we monitor those separately with like handheld probes or chemical testing to kind of like pH. You just use a strip, you dip it in, and you know right away what the pH is. So having a probe that sometimes reads wrong can be more scary sometimes than just like actually doing it by hand. This is our grow out facility page with our temperatures and our oxygens. So you'll notice that most of the tanks only have oxygen levels, and the 4 tanks in the middle have temperatures and oxygens. Those are our brood tanks. So it's more important for us to know the temperature on there because the temperature is what determines the temperature, along with the photo period, helps to determine when they're going to go into their spawning season. Again, which I'll talk about shortly. But this is just a picture of one of the graphs that you can look at. So this is just 4 days of temperature in our broodstock tanks, and you can kind of monitor the trend. You can see some of them are increasing as we bring them out of season. Some of them are decreasing as we bring them into season. And the blue and pink lines there are kind of stable. That's just variation in our well temperature as the year goes on. Now we can get into the stuff that I'm actually good at. Halibut production is a large majority of my life now. When I was in university, actually, one of the profs that I had a class with told me that I would never work with halibut in my life and not to worry about them. And here I am giving a presentation about it. So our broodstock is the only Atlantic halibut broodstock in North America. We feed them a diet of herring and mackerel, but we're working on developing a biosecure feed to ensure that the feed that we give them is free of pathogens. There's no naturally occurring viruses around PEI for Atlantic halibut. We've had a couple of people look into that from the University of Prince Edward Island to ensure that we're not really super concerned about that. But with fish coming in, the herring and the mackerel that we get come from anywhere from locally sourced to caught in like the North Atlantic around like Norway and then shipped over. So we are a little concerned about introducing a pathogen with the frozen herring and mackerel that we feed. Hopefully this the biosecure feed that we create will be beneficial. It's been really hard to convince our fish to take a sausage of pellet feed and like more like cooked fish and fish oil They don't seem to like it very much. I don't know if it's the taste, the texture, or like just the fact that it like doesn't look exceptionally fish-like. But they haven't really been interested so far in taking that. We have 3 separate egg seasons. We've got a season in February, August, and November. Those have been altered through the use of photoperiod lighting and temperature adjustments. And that gives us a constant, well, almost constant. We have a little bit of time off in the summer, around June and July. That was put into place by the company who used to have our brood stock before we did, so that they could have summers off. That gives us, yeah, like I said, like almost constant feed of eggs through the season. We have a 4th proposed season that'll kind of keep that stream running all year round. But we have some upgrades due to the facility for capacity before we can really stream into like 365 days of year of egg production. We harvest the eggs by hand. Here's a picture of one of our brood stock on the table for egg harvest. So you just massage the ovary and collect the eggs out of the vent. Milt is collected the same. We cryopreserve it in dewars so that we can hold on to a lot more milt over the course of a season. Just because it's really hard to like time one of the females going on to spawn and releasing her eggs and having like males ready at the time. Most of our broodstock are female. We have about 12 males that are in production right now producing milt throughout the year. So to kind of catch one of the males as a female is ready, the cryopreservation really gives us the freedom to fertilize everything that we get, which we also do in-house. I might have some more pictures on here. Yeah, this is just more broodstock. So some of the broodstock are wild caught. Some of them have been farm raised at other farms. A few of the ones that we just put in aren't necessarily producing eggs yet, but they are fish that have been around the facility since before me, but were hatched and raised in-house. I was gonna try to identify some of the fish in there for you because they all have a pit tag and a name, but I can't really like see anyone in particular that's impressive in that picture. The smallest broodstock we have is probably about 30 pounds, and that fish would be like 3.5 feet long. And then our largest, her name is Elliot. She's like 150 to 180 pounds, and she's probably about 6 to 6.5 feet long. So she's like significantly larger than me. It makes it pretty difficult to bring her up onto the broodstock table to get her eggs, because when they get up on the table out of the water, they're not always very happy. And she's got a lot more muscle than I do. So that is a little bit of a stress sometimes to do that. She usually takes about 2 or 3 people to handle, and they take handling very well compared to most fish. Like they won't usually, at this size, they won't usually go off feed after handling, and at the smaller sizes, it takes usually a day or 2, and then they'll come back on to feed. The egg system, so just like kind of an overview of what we do. husbandry wise, we have to take out the dead eggs every day. So the first few days of that is usually just like the unfertilized eggs. And then beyond that, we'll lose some eggs through the process. It's usually not like a very high number. We usually have pretty high success rates in actual like egg survival. Our hatch rates tend to be on the higher side of things, but some tanks, just for whatever reason, don't go off properly. Their hatching is asynchronous, or they're like some eggs just aren't ready to hatch when the other ones are. We disinfect them after the first week of them being in the system. We do that with a chemical called Parasan. It's peroxyacetic acid, I think, is the name of the compound. It's essentially just hydrogen peroxide and acetic acid mixed together. It's really nice because when it breaks down, it has limited, I say limited, there's no harmful components when it's broken down, so we can just like flush it down with our and put it through like our filtration before it goes in our effluent. Transfer happens at about 80 to 85 degree days, which is 2 weeks in human days. For anyone who's not aware, degree days is just the temperature-based development per day. So if the temperature is 5.5 degrees on day 1, you would get 5.5 degrees, and on day 2 you would be at 11 degrees. Working towards that 80 to 85 degree day mark for our hatching. We can actually inhibit the hatching enzymes from being produced with light. So this whole process is done in the dark, but around 10 to 12 days in, we turn an LED light on over the tank, and the cells that release the enzyme are inhibited by the light. So we can hold the cells off, allow the cells to develop, and then when we shut the light off, the enzyme is released in all the eggs at the same time. And that's what's kind of given us our very synchronous hatch, where all the eggs are kind of releasing that hatching enzyme at the same time to be released. And the transfer really just happens by hand. We take a beaker and all the fish kind of come up to the top in a little layer, and you just scoop them out and bring them over to our yolk silos. Couple of like small challenges in there are that the disinfection is a delicate process. Fish eggs tend to be a little bit delicate in general, so the chemicals that we use can be harsh sometimes on them if they're left in too long. It's important that you follow the SOP that we have in place, and then kind of make sure that you're being gentle with them. The eggs also need to be in constant suspension, so we have aeration in the tanks that keeps them circulating. This is a picture of some fertilized eggs. You can actually see the cells in the eggs are starting to divide. Most of them are just at their second cleavage at that point. This is just like a view under the microscope. The eggs are, I think, they're like 3 millimeters across when you look at them with your eyes. And this is just like a picture of some unfertilized eggs to contrast. It's like very obvious when you've done it right, which is really nice, because a tank of unfertilized eggs isn't going to do us any good, so we can just drain those if we didn't get the fertilization done properly. This check happens the day after they go into the tank, so we give them 24 hours to kind of start that cell division process, and before we like make any calls on whether or not we're going to keep them or not. Fertilization rate is like the first big hurdle that you have to like get through in the process. So in yolk, we have same thing, like the removal of dead larvae, which we just use. There's a drain at the bottom of the tank. Most of the fish will stay like neutrally buoyant in the water because we control the salinity. So the dead stuff all kind of ends up at the bottom. We can just purge it out into a net. And other than that, they really just take care of themselves. They're, stealth monitoring their buoyancy with the, like, as they kind of eventually they'll start to swim down towards the bottom. It's just a behavioral thing. And then closer to that 220 to 240 degree days, near the end of their time in yolk, they'll start to swim back up towards the surface. We use like flows to keep them from swimming straight down to the bottom. The transfer in this as well, they swim up to the top, we scoop them out and transfer them over. We can use a little bit of light to pull them in. Oh, that's just words, but I don't know what it said. Sorry. And yeah, so we have a few developmental issues that kind of show up. So this larvae has some edema in its yolk sac. You can see that there's like the yolk is on the top and there's like a little fluid buildup in the bottom. This one has what we call a punched yolk. So that's an issue with like the membrane around the yolk rather than fluid buildup in the cavity. This one has like its spine kind of messed up. We've seen it a few times. I'm not positive that it isn't just like somebody grabbing the fish weird with the pipette when we sample them. But it is something that we've noticed, and scoliosis is a problem further down the line, so I can't necessarily guarantee that it isn't a developmental issue. And then this one is kind of the silent killer of the fish. This is a gaping jaw. You can see roughly, I don't know if you guys can see my mouse, but there's like the mouth of the fish just next to the eye is like stuck wide open. So as that fish moves on into first feed, it can't actually like open its mouth to consume feed. And that any fish that has gaping jaw at this age will not make it through first feed. Once they've consumed their yolk, they'll starve. So this is like the last thing that you want to see when you're examining the development of your yolk larvae. I have a video for this one, which is really fun. We won't watch the whole thing because it's like a minute and a half long. But in first feed, we have our tank maintenance and cleaning tanks, like I talked about before. So tank maintenance is just kind of like keeping the tanks clean, scrubbing them because the clay kind of collects on the sides and on the bottom. We siphon out any kind of debris or any dead larvae that are in the tank. And they stay in there until about 50 days post first feed. So at this point, temperature is more important for growth, but the actual development of the fish is based more on time. So you'll see in the video that these fish, the larvae are actually a little bit different than when you see an adult. I'm using my hand because when they're larvae, they swim like a regular fish with one eye on either side of their head. And then as they develop and metamorphose, one eye will come across their head, migrate to the other side, and they'll start swimming on their side to like the more traditional flatfish that you're used to seeing. Atlantic halibut have I'm using my head to remember. Their eyes are on the left side, maybe. I can't remember. But the different species will have different, I don't want to say preference, but like evolutionary trend towards one side or the other for their eye migration. We do occasionally get fish where their eye migrates the opposite way. And developmentally, they're the same. They grow at the same rate. They taste the same. They just like flip the other way. And when it comes to if they ever make it to being a broodstock fish, it'll be more difficult for us to harvest their eggs because our table is kind of like L-shaped with like a wall for them to not swim right off the other edge. So the vent is on the opposite side of the fish to where we access it. Big challenge in first feed is our Vibrio alginolyticus. It's a Vibrio bacteria that's present in the cyst capsule of Artemia, which is the live feed that we provide them. It releases a neurotoxin, and it's basically once the bacteria gets established in a tank, there's not really a whole lot you can do to bring them back. Total gas pressure is also something that we control very heavily, but it can lead to like bubbles in the blood or in the eyes or in the brain. Here's our little video. We just put a GoPro down into the tank and you can see all the larvae. They're swimming the right way up. You can see Artemia in the guts. That's that little orange loop. They have no pigmentation at this point. Some of them are definitely more developed than others. You can see like as they develop, they get more like wide, rather than just being like skinny little guys. And all of these fish would be female because we use sex reversed males or sex reversed females for our milt. So all of the milt that we use actually has female genetics. So the cross always results in all female brood or all female stock, I guess. This just kind of allows for more control over the process. Males tend to grow slower and end up smaller. I guess we will watch the whole thing because I found things to talk about. But females just tend to be better for production and more important to have at the time of spawning because we don't have a way to cryopreserve the eggs just yet. But there are some companies that are working on that process. Oh, we're gonna play the video again. Oh. Artemia is our live feed. We buy the cysts in a can and put them in water to hydrate them. After hatching, we can separate the cyst debris from the hatched Artemia with magnets. That's a trademark system called SepArt. They magnetize the cysts, like the cyst wall, so that once the Artemia have hatched, we can flush the tank through a tube with some strong magnets in it, and it'll actually like pull the cyst debris and collect it on the magnets and allow the Artemia to flow out into a new vessel. We enrich them with a product called Orient 3. It's a high fatty acid, high protein diet that allows, it's basically we feed the Artemia so that when we feed the Artemia to the halibut, they're more beneficial. Nutritious is the word that I wanted to use there. The enrichment process takes about 20 hours and the whole process start to finish takes about 2 days. So you can see the picture on the right is an Artemia. They're a small brine shrimp, and you can see through their gut they are filled with fatty acid globules. And you can see the eye spot at the end. I think they're cute, but they're kind of terrifying, I guess, when you see them like close up like this. And this is something that we produce in like the tens to hundreds of millions per day. And I think that's like a small amount compared to some other farms that use this. We're currently developing a disinfection protocol to help control the Vibrio species that I talked about in the last slide. After first feed, they move on to the nursery and the grow out. So same kind of things like with husbandry, we have our regular tank maintenance. So there's just more tank to clean usually. And with the pellet feed being added, the waste and waste feed create quite a mess. We have a picture on the far right is our grading setup. So we grade by hand. You just look at a fish, decide if it's big or small, and then send it into one laundry basket or the other. And we put them into the tanks through that. Also, during the grading process, not in this picture, but we'll sometimes put a Floy tag, like just a little external tag on the fish to mark it for recruit selection. So that's a fish that's growing at an exceptional rate or exhibiting some kind of feature that we deem helpful for broodstock. And they'll kind of be tracked through their growth to see if their genetics are good to join our broodstock program. And then they stay in the nursery until they're about 150 to 200 grams, and then they go to the grow out and stay there until harvest. The two systems are very similar. The only real difference being that the grow out is on recirculating water and the nursery is all flow through. So the grow out has the additional challenge of the recirc system. Across the whole thing, we have eye picking that becomes a problem. Like if the fish don't feed enough, or if there's like if they're being underfed or they have the wrong feed size, they won't eat and they'll start picking at each other's eyes. I didn't put a picture of that in because it's kind of gross, but I do have a picture here of a fish with scoliosis. You can see that its tail is at wild angles. And that's usually based on like early nutrition. But there is a genetic factor to that as well. So here's our spotted wolffish. Right now we have 12,000 spotted wolffish that are about 6 months old on site. Yes. That's okay. Sorry about that. Yeah. Sorry. And then we have 66 that are 4 months older than that, which you'll see in this TikTok video that I took. The spotted wolffish don't require live feed as they age up, so they kind of get onto that high conversion pellet feed a lot quicker. They're very fast growing, but they're also cannibalistic. So 24-hour feeding is very beneficial to those fish. We also grade them very frequently, because if we leave small fish with big fish for too long, they'll just start eating each other. I heard that the industry standard cannibalism mortality factor, because you can't see a cannibalistic mortality in a tank, was at 1.50% from grade to grade. So about half the fish would get eaten in a tank at a time, but we're definitely down from that now. With feeding and regular grading, we've kind of minimalized the cannibalism. You don't see it very often, but it's kind of the beast you have to fight with them. In future, so around February, we'll get a transfer of wolffish broodstock from Quebec. I don't know how many exactly we're getting, but we're putting them in 6 tanks. I know that much. But we will have to do a full overhaul of this facility to create brood rooms and egg rooms that are biosecure from our halibut, just to keep the any pathogens that might cross over separate. The brood, like the broodstock plan and all of the SOPs and processes for that have already been established by the company that AMAR has been working with in Grand Riviere. So once they come in, it'll just be a matter of learning how to do that, and we'll be growing wolffish for food. Here's my little TikTok video, just showing the fish. If anyone's really big on TikTok, it has the Jaws theme playing in the background, but I just didn't put the sound in. They're relatively docile in the tanks, so like you can see, I picked that one up. I cleaned and disinfected my hand and then rinsed it in some ambient tank water that we have separate before I handled them, just to ensure that I didn't stress them out too much. But they are, like, most of the time they just sit around on the bottom and wait for food to come find them, unless there's some kind of distress that causes them to swim somewhere else. They really like to sit. You can see they're kind of in a little bit of a clump. When there's more fish in the tank, they clump up even more, and they'll even like feed in balls on occasion. They'll just like swim in a group and just like frenzy feed, which is something that I'm not used to with the halibut, because they don't really do that. It's been a really exciting experience starting a new species of fish. So we're on to my couple interesting opportunities that I've had. The first one is a PhD process by Ellie and Dr. Daniel Rouzant from Dalhousie. I think I said his last name right. He told me to just call him Daniel when he was here. With the help of Chris and Mallory, they're using DNA methylation to look for genetic markers in the halibut genome that can find a way to age them rather than using the otoliths like they would traditionally do. I have very limited knowledge as to how that process works, but I got to be a part of collecting the DNA with fin clippings because our facility provides the opportunity to examine the DNA of fish that have known ages to identify these markers. We had 80 to 100 fish at the time in the tank that they were looking at, where we knew the age of each one, spanning from fish that were spawned in 1999, so they'd be 23 years old at this point, up to fish as recent as 2018. So we gave them a nice wide spread of ages to kind of look at. And yeah, their hope is to be able to just take a fin clipping and determine the age of wild halibut without actually having to like dig into their ear bones. They're really nice people. It was a lot of fun to be a part of something so like above my head in science. It was very interesting to learn about. And the other big thing that we have is a feed trial that we ran last year, and we're kind of still in the process of continuing the trial right now. We're working in conjunction with a feed company in the States to trial a new weaning feed for halibut larvae. Results have been very promising so far. I found out today that they're actually like the results are confidential, so I can't talk about them. but you can see from this picture that it's pretty obvious that one of these samples is handling this a lot better than the other. So it's working really well. The same company and I are developing a protocol to feed disinfected frozen copepods to the larvae. So that would take the place of the Artemia, the live feed. Copepods are essentially the same kind of thing, just like a more broad term, and I think his are wild caught. But they're disinfected, so there's no bacterial contaminants introduced in the live feed, which is like our big problem right now. So if we can get that to kind of go off, it'll change the game pretty quick. The issue that I've found so far with that is that the halibut larvae really like to hunt off of movement, and dead frozen copepods don't move nearly enough for the fish to be interested in them. We tried the copepods once when they were at like 40 days post first feed, so they were very used to hunting by movement at the time. So I think if we had like a more gradual transition and also started earlier, we get a lot more success out of that. But yeah, thanks for being around. That's me in an amount of time. I rambled a bunch. I apologize, but yeah, that's what I got.
Cool. Thank you so much. That was great. Very informative. I know I knew nothing about fish or halibut aquaculture, so I really appreciate you sharing everything with us. I'm gonna open this up for any questions from anyone. Okay, we've got one inside Hampton Hall, so one sec.
Yep.
Right. Thank you, Ben. My name is Colin. First of all, I'd like to thank you for being anti-cannibal. You know, that's one controversial stance I take, is being very anti-cannibal. So I know I get a lot of crap for it, but I'm against cannibalism. So when it comes to genetic diversity, I'm thinking of the bigger farming techniques with lots of broodstock, and that'd be salmon and trout. And there's so many like commercial broodstocks that you could pick from, and different populations and streams, and you can really up your genetic diversity game. I know you're controlling for a lot of the diseases they might get with the land-based aquaculture, but is the company concerned about a lack of genetic diversity in these aquaculture fisheries, where there's such little being done? Like, you guys are the only halibut ones, so I mean, if you wanted more halibut, you just go on a commercial boat or something and up the diversity, or how does that work?
Yeah, that's exactly what we do. So we get a permit to go out during the regular halibut fishing season, and we will, like, as they pull up wild fish, we have somebody on the boat that'll kind of like identify a fish that might be better or worse. Well, I guess they would identify the better ones. We don't want to take worse fish for broodstock. But we do pull wild fish out from the water, which is a permit that we apply through the DFO to get. And we have 1, 2, 3, 4, 5 different sets of fish that were wild caught. We actually had a genetic profile done on our fish a few years back. And even within our facility, they found that there's actually 2 distinct populations of halibut within just the fish that we have. So that's like an interesting fact, I guess.
Yeah, cool. So I know there's the wolffish is a little bit newer. Are you guys doing the same kind of thing with the wolffish?
I would assume that we'll do a similar thing. We haven't actually done anything in-house with wolfish broodstock yet. But I do know that there's a lot of like wolfish that are captured through bycatch. I'm not sure if like if it tends to be more spotted wolfish or a different species that's getting pulled. But I assume that eventually we'll be doing a similar thing for them, yes.
Awesome, thank you.
You're very welcome. Thanks for your question.
We've got one question in chat, and I was about to ask this too. Who do you sell your halibut to?
Yeah. Hi Jordan. Haven't seen you in a while. Um, Jordan and I used to work together. So we sell our halibuts to a couple of local distributors. One of which burnt down this year, so not to them anymore. But there's a company in Charlottetown that we sell to, and they distribute from there as far as Montreal. We also sell a little bit direct to customer if the right people show up, or also to restaurants directly. We've had a couple chefs reach out to us to purchase farmed halibut so that they can like kind of see the difference. I don't find that there is one, but maybe somebody with a more distinguished palate would be able to tell the difference. Yeah.
Great. Thank you so much for this talk, and thank you everyone for joining us. I think we're a little bit over, but our next seminar will be November 4th at 1:30, where we'll hear from Sophia Montalvo. She's a climate scientist with the U.S. National Ice Center. So don't want to miss that. And guess that's it. Thank you, guys. Thank you, everyone.